Okay, hello everybody. We will wait just a couple of minutes and we'll start with the webinar. So hello to everybody who is joining us now. We'll wait just 1 minute more and we'll get started.
Hola, buenos días.
Hi, I'm Milton.
Hi, Vivian.
Buenos días.
Hi.
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 Hello, Nandini. Hi, Lilian.
Hi, Milton.
Hello, Milton.
Okay.
Hola, Catarina.
Yeah, Katerina
Hi,
Shubha.
Milton. Hi, Milton.
Yeah,
Yes.
Shubha has joined. Can you make her panelist?
All right. All right, so I think we can start now. So good morning, good afternoon, or good evening, depending on your time zone. Welcome to this new webinar, the 7th in the series of the Trevor Platt Science Foundation. As you know, this is an international organization dedicated to capacity building in science, research, and education related to our oceans, working towards stewardship of our planet for future generations. Today's webinar is one of the capacity building activities of the Foundation. And before introducing our speaker today, just a couple of instructions. As you will notice, your microphones are off. Please post the questions in the Q&A compartment, and we will get to them after the seminar. So today's speakers is Dr. Montserrat Aldunate Chinchón, who received her PhD in oceanography at the University of Concepcion in Chile. At present, she is a researcher at the Millennium Nucleus for Study of Deoxygenation in the Southeast Pacific Ocean, University of Concepcion. She has specialized in the study of marine microorganisms and phytoplankton and their relationship with the environment and their role in biogeochemical cycles. She teaches undergraduate and graduate students, participates in national and international cruises, and takes part in multidisciplinary research teams. Her work involves technical experience integrating flow cytometry, microalgae culturing, stable isotope experiments, and molecular biology. And today she will be talking to us about microscopic organisms in hypoxia, ecophysiological rates, and their role in the carbon cycle. So, Montse, the floor is yours.
Thank you very much, Vivian, for introducing me and also for the invitation. As Vivian said, today I'm going to talk about microscopic organisms in hypoxia and why in hypoxia, why it's important this, because some of you know that ocean is losing the oxygen. You can see here, this is a plot with the oxygen inventory and how this oxygen has been getting lower with the time and much lower in the deepest depths of the ocean where we don't have too much mixing with the atmosphere. And also, we have been seeing this in the observations that they say that we are losing about 2% of the oxygen in the global ocean. Some IPCC models show a lower percentage, but maybe because don't have enough information or of lack of the of measurements in different oceans. And how deoxygenation is impacting in the ocean can be impacting in the diversity and physiology of marine communities, also in the marine biogeochemical cycles, in the earth's greenhouse gas fluxes. So it's impacting in several ways. And also it's impacting in natural places in the ocean that is naturally with low oxygen, like the oxygen minimum zones. And you can see here, this is the Eastern Tropical North Pacific, Eastern Tropical South Pacific, the Eastern Tropical South Atlantic, and the Arabian Sea. And you can see here very low values of oxygen, and actually in some places has been renamed like anoxic marine zones because they experience total anoxia. So that means no oxygen at all. And what we can see or what are the profiles that we usually see in that kind of zones, we see first an oxygen, anoxic layer in surface layer, like 100 and 500 meters depth. Then plenty of nutrients, also with this special attribute that is the NO2 accumulation in these anoxic marine zones. Some instruments or some sensors, the sensors that we usually use in oceanography have a detection limit of 2 micromolars, so they cannot measure anoxia. So when we see NO2, this accumulation in these anoxic marine zones is like a proxy of anoxia, because nitrite has been using like electron acceptor because there is not oxygen, so microbes use NO3 and accumulates in these zones NO2. And also a high microbial abundance and a very characteristic profile is in the chlorophyll chlorophyll A, that we observe this secondary chlorophyll maximum at the top of this anoxic layer. And this secondary chlorophyll maximum is just below the euphotic layer. Less than 1% of light are experiencing these microorganisms, with Prochlorococcus being the most important photosynthetic organisms and contributing at least the 90% of this secondary chlorophyll maximum. And these Prochlorococcus, they are not, or they are a special lineage of Prochlorococcus that has been only found in these anoxic marine zones. So what happened with all of this? What happened with the oxygen or the deoxygenation? What happened also with the OMZs? The OMZs, or the oxygen minimum zones, are expanding. That are expanding locally, that are expanding or getting less oxygen over the ocean, but also they are expanding to surface layers, and also in to deepest layers. So, and why they are expanding? Because the global warming, the upper ocean layer is warming. That means that is the capability to dissolve oxygen is lower. Also, this warming enhances stratification. That means that there is less deep water formation, reducing the oxygen supply and expanding these oxygen minimum zones, reducing the habitat to shallower depths, reducing the habitat for fishes, zooplankton, but not for microbes, and also with the anthropogenic and natural inputs of nutrients. Also, there is a variable biomass production that is remineralized and consuming oxygen. So we don't know what is going to happen to phytoplankton or bacterioplankton with this expansion. Maybe, or we have been seeing that also these oxygen minimum zones is reaching the photic zone. So what happened with the phytoplankton? Phytoplankton inhabits layers, plenty of oxygen with sunlight, and they do photosynthesis. They take in CO2 plus water. They take energy from the sun to produce organic matter for their own machinery, metabolic machinery, and producing oxygen. But when we analyze these profiles of phytoplankton, we observe that in the oxygenated surface layer, there is phytoplankton of various sizes, eukaryotic plus prokaryotic phytoplankton. We can find diatoms, dinoflagellates, coccolithophores, cyanobacteria. But when the oxygen, here in the oxygen line, when the oxygen goes down, also we saw this characteristic profile where chlorophyll or fluorescence is also decreasing, reaching a point in the oxygen line with a chlorophyll minimum. And then when the oxygen, or when reached the anoxic layer, we saw this, we have been seeing this secondary chlorophyll maximum. That is mainly composed by prokaryotic phytoplankton and the 90% Prochlorococcus. But the question, why is there a chlorophyll minimum when oxygen concentration decrease oxygen, decrease chlorophyll? Why happened this minimum? And the question was, do eukaryotic phytoplankton need oxygen to survive? We know that phytoplankton are estimated to contribute about half of the global phototrophic primary production and are therefore the major contributors to oxygen on Earth. Why will a lack of oxygen affect them? That is the question that we have. And this is a first experiment or work that we did with Edson Piscoya, a master student in oceanography here in Universidad de Concepción, University of Concepción, Peter Fondasso, and Cristian Vargas. And we take samples from northern Chile, where we have this very extent and intense oxygen minimum zone or anoxic marine zones. And we did this transect and trying to figure out what environmental factors could control the vertical distribution of picoplankton associated with the OMZ of northern Chile. And with all these variables like temperature, oxygen, chlorophyll, nutrients, carbon system parameters, salinity, etc., we determined that temperature, oxygen, and carbonate chemistry were the principal parameters explaining the variability of this phytoplankton and the distribution. Sorry. We also, in this work, did one experiment using Imantonia, that is a strain that was isolated from the same area in the northern Chile, and exposing this Imantonia to control parameters in pH, in oxygen and light, and also in OMZ parameters, that means 7.5 of pH, hypoxic concentrations of oxygen, and light enough to do photosynthesis. And we saw that we did this experiment in 6 days, and as expected in the control, Imantonia grows, or the fluorescence increase, the pH also increased because Imantonia is fixing carbon and also producing oxygen, is what we expect. But when we expose that strain to OMZ conditions, OMZ treatment, Imantonia not survive, and you can see the same or related parameters with the pH, that they are not too much different between 0 and 6 days, after 6 days. The same for oxygen. So we think, okay, oxygen seems to be like a parameter that is affecting phytoplankton. So we work in this paper with Jane Wong, John Rayburn, and Osvaldo Ulloa, Peter Fondasso, and the other collaborators that you can see there, in a hypothesis that is that phytoplankton require oxygen to survive. So what we did, we analyzed 147 published data profiles from the Eastern Tropical, North and South Pacific. These profiles, we analyze flow cytometry profiles, hydrographic profiles, CTD, like is CTD-O and fluorescence and PAR, and we trying to explore different hypotheses, trying to explain this minimum of chlorophyll, trying to explain why this there is this minimum. So first, the first hypothesis was maybe grazing, but there is not evidence of protists grazing at the chlorophyll minimum. In addition, as is very low oxygen, most of the protists needs oxygen to survive, so we cannot find this evidence in there. Other hypothesis was irradiance, but the fluorescence minimum is over the 1% of irradiance, so they have enough light to photosynthesize. Also, Prochlorococcus, the lineages of Prochlorococcus inhabiting the OMZs, the top of the anoxic layer, they are photosynthesizing. So we discard that that hypothesis, and the last hypothesis was oxygen inhibition. Here you can see the concentration of oxygen in the minimum of chlorophyll to different cruces. Those are the names of different cruces, and those are the concentration of oxygen for the these chlorophyll minimums, and we calculate that the chlorophyll minimum occur at the average of 3.5 micromolar of oxygen. That is very, very, very low, and 90% of this chlorophyll minimum was in between 2.1 and 21.3 micromolars of oxygen. So we saw that there are oxygen-dependent metabolisms like photorespiration and water-water cycles that have low affinities and could explain these patterns. We know that these metabolic pathways that usually used for phytoplankton and that need more oxygen has been inhibited. Sorry. So we propose a model. This panel, the panel A, is the fluorescence that is observed. Actually, this is station 11 in the RB 1603 cruise, that we observe this primary chlorophyll maximum and then this secondary chlorophyll maximum, and below 100 meters depth is anoxic, and we made this simple model, and you can see here in orange, this is the growth of phytoplankton only based on light, and this when we apply in green, when we apply an inhibition of oxygen for oxygen, this represents the Prochlorococcus OMZ lineages that are inhibited in the surface because the oxygen, and here the oxygen-dependent profile that is because the photorespiration and water-water cycle. And when we merged this profile, we found that the missing photosynthesis is about 27% in this profile. In other profiles, is ranging between 22% to 27%. So the model, when we take in account the light, but also the oxygen requirements that were included in this model, reproduce the observed pattern very well, actually. So how might the expansion of oxygen-deficient waters affect the illuminated surface waters? Will it stimulate the secondary chlorophyll maximum? Will it negatively affect the surface chlorophyll maximum? What will be its biogeochemical consequences? Now I'm going to show you some experiments that made by Fabian Cortés, master student in oceanography. This is part of his thesis. And his idea is trying to figure out how the phytoplankton community is affected to hypoxia, but in experiments. He tried to concentrate phytoplankton, pumping a lot of water, but and exposing this concentrated phytoplankton to surface water, oxygenated water, and also to OMZ water, but the result has been inconclusive. But he also tried with different strains of phytoplankton. These are results for Hephirocapsa huxley, or Emiliana huxley, that is a coccolithophore, and he exposed this strain to normoxic water taken taken directly from the ocean. He did these experiments on board the Falcor 2, a Falcor 2 cruise, and exposing also with low oxygen waters that comes from the oxygen minimum zone. And he saw that in vivo fluorescence, in the case of the normoxic treatment, was increasing during the days. He did this experiment until 22 hours. But in the low oxygen treatment, there were not changes or changes among the days. The same trend was shown in this in the abundance of Hephirocapsa huxley, with increasing of the cell abundance in the normoxic treatment and increasing at the beginning, but stabilizing in the next days. And this is for photosynthetic efficiency and showing the same trend. In the case of Thalassosira, that is a diatom, the trend was different. Here you can see the fluorescence for that fluorescence was increasing, passing the days. The abundance showed the same trend, and the photosynthetic efficiency showed the same trend for both treatments, normoxic and low oxygen. Actually, this low oxygen is 40 micromolars of oxygen, very hypoxic concentrations. So oxygen availability affect different type of phytoplankton differentially. So the community response should depend of the community composition. That is the one of our conclusions. We know that we need more experiments because that was based in only two strains, but now we're trying to concentrate better better the phytoplankton, maybe with a phytoplankton net. We are trying to also seeing with different kind of on other models. So that is for the phytoplankton. And what happened with bacterioplankton? Bacterioplankton in the OMZ live in darkness and hypoxia or anoxia. And in one study of Christian Vargas in 2021, I think, sorry, I don't put the year in there, he showed that for the same place, also of Iquique, when we found low primary production at surface, but still low oxygen in the subsurface depths, there are changes in the delta 13, carbon 13 of the particulate organic carbon. Both places, this is in 2015, low primary production, high primary production for 2018, in both low oxygen, in both NO2 accumulation, but the difference was in the delta carbon 13 of POC. A deplete value of carbon 13 POC is a proxy of chemotrophic carbon fixation, and enriched values are proxy of heterotrophy. But that was only one observation, and with that observation is that I take that observation, I made this postdoctoral project that is the impact of the photosynthetic primary productivity over the carbonate chemistry and carbon fixation in these subsurface oxygen depleted waters. The idea is to try to compare different regimens of primary production, surface primary production, to see how changes the carbon fixation in OMZ. So I take samples from March and August, trying to, in here, also in the north of Chile, of Antofagasta, this is Mejillones Bay, and trying to, I proposed this to different opposite seasons, to trying to catch these differences in the primary production, in the surface primary production. But when we see this, how it was the oceanographic context, when I took the samples, this is the year 2022, and this is the year 2023. And the chlorophyll A at the surface was very similar in March and in August. So that are like my high productivity season. And in 2023, the summer and winter have very low productivity at surface. So I took this carbonate system parameters, and here are the March and August 2022 with high productivity. Those are the profiles of carbon chemistry. Always in black, the oxygen content with very low concentrations below 30 meters depth, and with very similar profiles in the pH and the PCO2, DIC, and total alkalinity, with very low pH values in the deepest depths in the oxygen minimum zone, high values of PCO2, high values of DIC, very similar between March and August 2022. And this was the low productivity period of season in August 2023 with a water column, mixed water column. They don't have very low oxygen content. He does not reach hypoxia, and very different profile. We also did these carbon fixation rates. We took water from the surface to points in the oxycline and the OMZ, and we put in bottles. We inject bicarbonate 13, and we incubate this in situ over 12 or 16 hours. And those are the results of this high productivity period and low productivity period. And during the period of higher productivity, there was a greater surface carbon fixation, but also I'm going to show you that in these panels below, you can see only the carbon fixation in dark, this representing approximately 11% of the total carbon fixation. That is not always considered when we talk about carbon fixation in the ocean. So it's a great percentage, but the bad thing is that with these 3 seasons, it's difficult to have a pattern. So we need to include more measurements. We have more measurements, but needs to be processed, the samples, to have a better understanding of how this works. So finally, some take-home messages. Temperature, oxygen, and carbonate chemistry influence phytoplankton distribution in oxygen minimum zones. The oxygen availability affects different depending of the phytoplankton strain, depending of the kind of phytoplankton. So the community response should depend of the community composition. The model, the Wang et al. Model, suggests that OMZ expansion could result in a significant inhibition of photosynthesis that is modifying the biogeochemical cycles. Can be losing like 22 to 27% of photosynthesis. Carbon fixation rates show a predominance of photosynthesis at the surface, of course. However, dark fixation rates represent around 11% at the surface. And the knowledge about the responses of phytoplankton and bacterioplankton to expanding low oxygen condition is still very limited. And this is the last. What is next? Evaluate the response of natural phytoplankton communities maybe using other methodologies to concentrate phytoplankton, identify other possible hypotheses for chlorophyll minimum, since OMZ water not only differ in oxygen from the surface water, analyze field observation of photosynthesis versus irradiance relationship under controlled oxygen conditions, analyze more carbon fixation rates to have to expand our data set that will allow us to better identify patterns. And I don't say it before, but we also have molecular samples that now we are going to start processing to identify the bacterial groups involved and trying to associate these carbon fixation rates with what bacteria are doing that, what metabolic pathways are using, are the changes in the expression of these pathways. And here part of the group, Peter Fondazo, Fabián Cortés, Sibyl Améstica, Emilio Espinosa, aquí Osvaldo Ulloa, por supuesto, el grupo aquí que me ayudó a hacer esta system, this group, sorry, I changed to Spanish, this group that helped me to make these chambers to PI curves, and the people in Mejillones in the north of Chile also helping me in the carbon fixation rates. And thank you very much. Thank you for your attention. Gracias.
Thank you very much, Montse, for your very interesting talk. And so now we'll see if we have questions here in the Q&A box. I don't see anything, but Katarina, maybe you have some that were posted on the chat? Well, I don't know if...
No, Vivian, we haven't.
Okay.
No one has asked any questions.
No questions. Maybe you were so clear.
Maybe my pronunciation was not so good. Sorry for that.
No. Well, at least for me, we have the same accent, so it was fine. But, well, I will take advantage. Maybe somebody will get inspired to have some questions, but I have one that occurred to me during your talk. The first experiments that you showed regarding the response of different strains of phytoplankton to the oxygen depletion. One was with Imantonia, and that was the growth was reduced. But on the other hand, Thalassiosira seemed to do fine with the oxygen depletion. That was sounds strange to me. Do you have anything more to add to that?
Yeah, I think that maybe they have different strategies. That is not the same like the animals that need oxygen. Also, they have different critical pressure of oxygens to perform their metabolisms. We think that that also phytoplankton have different thresholds to and strategies to avoid this problem. And maybe in coccolithophores, they need very, very, very low, Thalassiosira need very, very low oxygen content to change, because in Thalassiosira we did this experiment in like 40 micromolars of oxygen, but the minimum of chlorophyll, the average is like 2.5 micromolar of oxygen. The 90%, the maximum is like 20 micromolar of oxygen. So maybe if we go down the concentration more, maybe we expect that decrease in the growing.
Okay. Thanks, Montse. Here, there is a question in the chat. I'm not sure if it relates to your talk, but let's see. It says, do you have any model to predict HABs?
Not really, but now we are going to go in a cruise that we are going to take some measurements of different, the diversity of phytoplankton, but also measurements of different toxins. So maybe, yeah, so maybe we can answer this the next year. I don't know, but we are working on that.
Looking into that. Great. Yes. Okay. Okay, I will ask you something else. You mentioned at the beginning of your talk that this second chlorophyll maximum that was within the oxygen minimum zone was usually due to a special strain of Prochlorococcus.
Yes.
I understand that it's very difficult to culture it, but because I was wondering if you could do the same experiment that you mentioned with Imantonia and Thalassius aura with Prochlorococcus.
Yeah, actually, I did some experiments, but not in culture because it's not culturing yet. But I did some experiments to trying to see what sources of nitrogen is using this Prochlorococcus, because how they are in a place that they don't have too many competitors and also have a lot of NO3, and they have the gene to do that. We hope or expect that they are doing things in there, but we are not being exposing now Prochlorococcus. We have like 10% of the total abundance are from Prochlorococcus. So we did some experiments using the natural community or population, but not in culture.
Okay. Thanks, Monte. Now we have a question by Shuba here. She says, Montserrat, thank you for a very interesting presentation. I am curious to know if similar vertical structure in phytoplankton is also observed in other minimum oxygen minimum zones.
Yeah, that is in the cruises that I was participating. They are in the Eastern Tropical North and South Pacific, but also in the Arabian Sea. I don't has not been in the Arabian Sea, but I know that exists. They present this secondary chlorophyll maximum, very similar in the top of the anoxic layer. But the observation that I made is that in the Isotropical North Pacific, the secondary chlorophyll maximum is more intense. In the Isotropical North Pacific, sometimes the secondary chlorophyll maximum is very similar in intensity like the primary chlorophyll maximum. Here, of Chile, of Peru, the secondary chlorophyll maximum is not too big, but it's always present. And in Arabian Sea, is is similar, has been seeing some profiles very similar to the tropical South Pacific. So, but they are the three places that we are seeing this feature, this profile, this kind of profile.
Okay, thank you. Okay, here there is another question in the Q&A. I don't know the name because it's logged with with Motorola, but well, let's see who's the question. Do you think there is a change in the intensity of oxygen depletion zone in the global ocean? And second, is there any shift in oxygen depletion zone in the near future?
Yeah, actually the deoxygenation is a global problem or change. Has been seen in all the oceans. So because the warming is affecting, it's a global process. Some locally, the oxygen depletion also has been found, like the dead zones, but that is related with local process like high nutrient supply to the ocean, but global warming is affecting all the oceans. But in the oxygen minimum zones, like they are naturally with low oxygen, they are being more intense. That means like maybe in the past at 100 meters, the values were like 50 micromolars, for saying something. And now that is changing and being more intense. So the concentration of oxygen is getting lower. And the models and the observations show that this oxygen is decreasing with the time, with the observations, but also the projections, and showing that.
Okay, thanks, Montse. We have another question in the Q&A. This is from Antonella Decian from Argentina. Thank you so much for the presentation. I think it's going to be very interesting when you can sample the community composition at that minimum layer. Was the chlorophyll minimum also correlated with the nitrate minimum? I wonder if that condition was also affecting the decrease in chlorophyll. I also wanted to ask how shallow this hypoxic layer has been recorded.
About how shallow, I saw this, the shallower, like 40 meters, very shallow. And yeah, actually they are in the minimum of chlorophyll, they are a lot of nutrients. We can find a lot of NO3 at least. Also NO2, but NO2 is very low in all the ocean and accumulates there in below. But I think that it's not a thing, a nutrient thing, because they are nutrients in there, enough nutrients. And we are taking some samples from the, we filter a lot of water for the chlorophyll minimum to trying to catch molecular samples. Now we are processing that samples to RNA to see the different expression, to trying to figure out what if the expression tell us something about that.
Okay, thanks, Montse. Well, Well, I don't see any more questions in the Q&A or in the chat. So I don't know if there are more questions from the panel or comments. Well, if
Only to
not,
thank Montserrat for
Shuba had
a
a question. Shuba, yeah.
Shuba, go ahead.
only to say thank you very much for a really interesting presentation. Very clear, very interesting. It's a
Thank
big
you, and
problem.
I hope to show more results maybe in the next opportunity.
Okay, thank you.
Thank you for inviting me.
Thank you.
Milton, go ahead.
I was saying that Shuba had a question. Indeed, I have maybe a question out of your main topic here, but have your experiments with carbon isotopes were with carbon 13 or 14?
13.
13.
13 because the most of the ships don't allow anymore carbon 14. We know that is better to carbon 14 because we need less volumes. That means more replicates, a lot of things better, but we are trying to do this clean and we need more water to do that. We use like 1.2 liter bottles to do that, and we are in the we need a better experiment. It's like 5 liters a bottle, but it's not easier to do that on board a ship or we don't have enough space and water. But in the with more volume will be better.
So thank you. I mean, I also find it a very interesting presentation, very didactic, well illustrated. So I learned a lot. So those will be my comments, Vivian.
Okay, thank you. Thank you, Shuba and Milton, Nandini, everybody. So thanks to our speaker today, Montse, for your very interesting talk and for participating. And to all the foundation members, students, and other colleagues joining us today. And especially to the Earth Center and Katarina for hosting the webinar as usual. And so we'll meet together in the next webinar coming soon. Thank you, everybody.
Thank you, Vivian. Thank you, Katrina.
Everybody,
Katarina,
thank you.
Vivian.
Bye. Thank you.
Bye.
Bye. Bye, everybody.
bye. Thank you.
Bye.